Hello, science fans. Bill Nye here. Today, we're shrinking down to explore the world of cell biology, the building blocks of all life. Every living thing, from bacteria to blue whales, is made of cells. Cells are the tiny engines that power our existence. Understanding cells means understanding life itself. The study of cells began in the 1600s, when Robert Hooke first saw tiny compartments in cork and called them cells. Soon after, Antony van Leeuwenhoek discovered living single-celled organisms, opening up a whole new universe. Everything your body does, digesting, moving, thinking, happens because of cells. By studying them, we learn how our bodies work and how to fight disease. Cells are complex, organized factories, and we now have tools to see their tiniest parts, even their DNA. So let's dive in and explore the amazing world of the cell. All living things are made of cells, but not all cells are created equal. In fact, cells come in two main types, prokaryotes and eukaryotes. This fundamental division shapes the diversity of life on Earth, from the tiniest bacteria to the largest animals and plants. Prokaryotes, like bacteria and archaea, are incredibly simple and ancient forms of life. They've been around for billions of years, thriving in environments from deep-sea vents to your own skin. Their DNA floats freely in a region called the nucleoid. There's no nucleus and no complex compartments inside. Everything a prokaryote needs to survive is packed into one open space. Eukaryotes, on the other hand, have a true nucleus that securely stores their DNA, keeping it separate from the rest of the cell. Their cells are packed with specialized organelles, tiny structures that each perform a unique job, like energy production, waste removal, or building proteins. Plants, animals, fungi, and protists are all made of eukaryotic cells. This group includes everything from towering trees to microscopic amoebas. Think of prokaryotes as a studio apartment. Everything happens in one open space, with no walls or separate rooms. Eukaryotes are more like a house with many rooms, each designed for a specific purpose. This organization allows for greater complexity and efficiency. Because of their simple structure, prokaryotes are always single-celled organisms. They reproduce quickly and adapt to new environments with ease. Eukaryotes, however, can exist as single-celled organisms or join together to form complex multicellular life, like humans, mushrooms, or oak trees. The key difference is this: prokaryotes have no nucleus or membrane-bound organelles, while eukaryotes have both, giving them a huge advantage in complexity and function. Understanding this split is the foundation of cell biology. It helps us make sense of how life evolved and how cells work together to build living things. It's the first step to unlocking the secrets of life, from the simplest microbe to the most complex animal. Now that you know the basics, are you ready to meet the cell's inner team and discover what makes each part so special? Let's go. Let's zoom inside a eukaryotic cell and meet the organelles, the cell's specialized workers. The nucleus is the control center, storing DNA and directing cell activities. Mitochondria are the powerhouses, converting food into energy, ATP, through cellular respiration. Ribosomes are protein factories, building proteins from DNA instructions. Some ribosomes float freely, others attach to the rough endoplasmic reticulum, ER, which helps make and modify proteins. The smooth ER makes lipids and detoxifies substances. The Golgi apparatus is the cell's post office, packaging and shipping proteins and lipids. Lysosomes are the cleanup crew, breaking down waste and old cell parts. Each organelle has a specific job, keeping the cell running smoothly. Together, they form a busy, efficient factory. Without these organelles, complex life wouldn't be possible. That's the magic of the eukaryotic cell. Next, let's look at the cell's boundaries. Every living cell, whether it's part of a plant, animal, or even a single-celled organism, is surrounded by a plasma membrane. This remarkable structure acts as a flexible, selective barrier and is made up of a double layer of phospholipids, known as the phospholipid bilayer. The plasma membrane is not just a passive boundary. It actively controls what enters and leaves the cell, allowing essential nutrients and water in, while keeping out harmful substances and waste products. This selective permeability is vital for the cell's survival. Embedded within the membrane are specialized proteins. These proteins serve as channels and pumps, moving substances in and out, and as ID tags that help cells recognize each other. This makes the membrane dynamic, constantly adapting to the cell's needs and environment. Scientists call this the fluid mosaic model because the membrane is like a shifting sea of molecules, with proteins floating within the lipid bilayer. In addition to the plasma membrane, Some cells, like those of plants, fungi, and many bacteria, have an extra outer layer called the cell wall. The cell wall is rigid and incredibly strong. In plants, it's made of cellulose fibers woven together, giving the cell shape, support, and extra protection against physical stress and pathogens. Animal cells, on the other hand, don't have a cell wall. They rely solely on the flexible plasma membrane, which allows for a greater range of movement and interaction with their environment. You can think of the plasma membrane as the cell's gatekeeper, carefully monitoring traffic, while the cell wall acts like a fortress, providing strength and defense. Plant cells have both structures, working together to give them both control over their internal environment and the rigidity needed to grow tall and strong. These boundaries are absolutely essential for life. They help maintain the delicate balance of conditions inside the cell, which is necessary for all cellular processes. By keeping the cell's internal world stable and safe, the plasma membrane and cell wall allow life to thrive in a constantly changing environment. Without these protective boundaries, cells would lose their contents, be unable to regulate their chemistry, and life as we know it simply wouldn't exist. Now that we understand how cells protect themselves, let's explore how they connect and communicate with each other to form tissues and entire organisms. In multicellular organisms, cells must connect and communicate to work as a team. Cell junctions are the connections, like handshakes, between cells. Tight junctions seal cells together, creating waterproof barriers in places like your skin and intestines. Desmosomes act as strong anchors, holding cells together in tissues that face stress, like skin and heart muscle. Gap junctions are channels that let small molecules and signals pass directly between cells, allowing coordination, especially in the heart. In plants, plasmodesmata connect cells through the rigid cell wall, sharing water and nutrients. These connections let cells share information, resources, and strength. Without them, tissues couldn't function as a unit. Cell junctions are the glue and the communication lines of life. That's teamwork at the cellular level. Next, let's see how cells grow and divide. Every living thing, from the tiniest bacterium to the largest animal, is made up of cells. These cells don't just sit still. They're constantly growing, working, and dividing. This process is called the cell cycle, a repeating series of steps that allows life to continue and thrive. Understanding the cell cycle helps us see how our bodies develop, heal, and even how diseases like cancer can arise when things go wrong. Most of a cell's life is spent in interphase. During this phase, the cell grows, carries out its normal functions, and prepares for division by copying its DNA. Interphase is like the cell's everyday life, where it does its job and gets ready for the big event, cell division. When the cell is ready, it enters mitosis. Mitosis is the process where the cell's nucleus divides, ensuring that each new cell gets an exact copy of the genetic material. This is crucial for making sure our bodies function properly, as each new cell needs the right instructions to do its job. Mitosis happens in 4 main stages: prophase, metaphase, anaphase, and telophase. Each stage has a specific role, from condensing chromosomes to lining them up and finally pulling them apart into 2 sets. After mitosis, the cell isn't quite finished. Cytokinesis is the final step, where the cell's cytoplasm divides, creating 2 completely separate, identical daughter cells. These new cells can now start their own cycles. This cycle of growth and division is how we grow from a single cell into a complex organism, how we heal wounds, and how we replace old or damaged cells every day. But not all cells divide the same way. To make sex cells, sperm and eggs, our bodies use a special process called meiosis. Meiosis is different from mitosis. It reduces the chromosome number by half and shuffles genetic material, resulting in 4 unique cells. This genetic shuffling is what makes each person unique. The diversity created by meiosis explains why siblings can look so different from each other. This variation is also the driving force behind evolution and adaptation in all living things. In summary, mitosis produces identical cells for growth and repair, while meiosis creates diversity for reproduction. Both processes are vital for life's continuity. Without these processes, life as we know it wouldn't exist. Every cell in your body is a result of countless cycles of mitosis and, for reproductive cells, meiosis. The cell cycle is the engine of life, driving growth, healing, and reproduction, one division at a time. Next, let's explore how substances move in and out of cells, keeping them alive and functioning. The plasma membrane controls what enters and exits the cell. Passive transport moves molecules from high to low concentration, no energy needed. This includes diffusion, like oxygen, osmosis, water, and facilitated diffusion, using protein channels. Active transport moves molecules against the gradient, using energy, ATP, like the sodium-potassium pump in nerve cells. For large particles, cells use endocytosis, bringing in, and exocytosis, sending out, wrapping materials in vesicles. These processes let cells import nutrients, export waste, and communicate with their environment. Without them, cells couldn't survive or interact. The membrane is more than a barrier. It's a busy, selective gateway. That's how cells manage their world. Next, let's talk about cell death and renewal. Stay tuned. Not all cells live forever. Sometimes they self-destruct in a controlled way called apoptosis. Apoptosis is essential for development, removing damaged or dangerous cells and preventing disease. It's a clean process, avoiding harm to surrounding tissue. When apoptosis fails, diseases like cancer can result. On the flip side, stem cells are unspecialized cells with the power to become many cell types. Embryonic stem cells can become almost any cell. Adult stem cells help repair tissues. Stem cells offer hope for treating injuries and diseases through regenerative medicine. The balance of cell death and renewal keeps us healthy. It's a cycle of life, death, and new beginnings at the cellular level. Now, how do we study these tiny worlds? How do we explore the cell's hidden world? With powerful tools. The light microscope lets us see living cells and their shapes. Electron microscopes reveal tiny organelles in stunning detail. Stains and fluorescent tags highlight specific cell parts, making them glow for study. Cell culture lets scientists grow cells in the lab to test drugs and study diseases. Molecular biology tools, like PCR and DNA sequencing, let us read and manipulate genetic material. By combining these techniques, we uncover the secrets of how cells work. Cell biology is a universe of discovery, happening right now. Thanks for joining this microscopic adventure. Keep exploring, because science rules. See you next time.